This is the final prep for lecture 16, and I'm kind of thinking that both lectures 15 and 16 might go a little bit longer, so we may spread them over 3 lecture periods. We'll just see how things go when the actual lectures are given. And just try to be flexible as we go through. So we finished off talking about cases where you can map 3 linked genes with a trihybrid test cross. And you can do any kind of trihybrid cross this way. And I asked you to give some thought to what would happen, what kind of pattern would you expect to see if you had 2 genes that were linked in your trihybrid, but one was on a different chromosome, so it was assorting independently. And then what would you expect to see if all 3 were sorting independently? And hopefully you gave that some thought. And we'll look at those patterns, and we'll finish off by taking a look at the X-linked, the case of 3 genes on the X chromosome, and how you would go about doing that kind of mapping. Okay, so here's another 3-point cross, and in this case, notice these dots, which I will point out frequently because I just want you to get right burrowed into your mind that those dots mean we don't know the linkage relationship between the genes. And we make a trihybrid. And we do a test cross. And we see this kind of a pattern. Okay. And in this pattern, we have 4 large categories. And we have 4 small categories. So when I see a pattern like this, my first thought is that these are non-crossover and these are crossover. Okay, but that's not sufficient. I still have to figure out which are the linked genes and which genes are not linked. And so I have to look at the data a little bit more carefully. So I'm looking at the genotypes here, mutant for all 3, wild type for all 3. Mutant for scute and echinus, and wild type for scute and echinus. So I'm seeing something right here that I would say is a pattern, and that is that this category and this category are the same, and this are the same. So really, for scute and echinus, those are the only 2 categories we're seeing. And so those must be the genes that are linked. But scute and echinus mutant are sometimes associated with the mutant for vestigial and are sometimes associated with the wild type for vestigial. And we see those equally likely. And when the 2 wild types are together for scute and echinus, half of them are associated with the mutant vestigial and the other half with the wild type. So I'm going to suggest that by looking at this pattern alone, you can figure out that scute and echinus are together on a chromosome, and that vestigial is on a different chromosome. Now, we still have to do the mapping. And we'll use these data because in these categories here you can see that the wild type scute is associated with a mutant echinus and vice versa. So these are going to be our recombinants. And I have the analysis on the next slide. So that was all the thinking, and then we just figure out the rest of it, right? So scute and echinus we looked at first. The recombinants are this one, this one, this one, and this one. And so we take these guys, 26, 42, 56, out of 1,008. So that gives us 5.6%. So that tells us that those are 5.6 map units apart. And then you can just logically see that each of these categories, the scute, wild type, and echinus mutant, is equally likely to be associated with the mutant for vestigial or the wild type for vestigial. And the same with all the other categories. Each of these categories for skewed and echinus is equally likely to be seen with vestigial wild type or not. But if you want to really convince yourself, you can take a look at skewed and vestigial. So that's a parental, that's a parental, that's a crossover, that's a crossover. This one is parental, parental, and that's a crossover, and that's a crossover. And so if you add them all together, you'll see that it comes to like 50%. And so if we see an R value of 50%, then we will conclude that those genes are not linked. And for your own sake, you should repeat that with echinus and vestigial, and you will find that you get the same. The exact numbers you use are slightly different, but you're going to get something that looks like it's around 50 map units, and that would mean they're actually not linked to each other. So that's a quick run through that, and this is me animating everything. But I actually just like to use the pen instead. I'll use these animations in the lecture though, so I'm going to leave those in. So, okay, so, and hopefully you thought of, if the 3 genes are not linked at all of them are on separate chromosomes, what the pattern would look like. I'll probably have a question about that in the lecture as well, to make sure that you've thought about it, and if you haven't, that you along with the rest of the class figure out the answer. So what about mapping genes on the X chromosome? So in some ways it can be even easier. So suppose you do a P cross with females that have a mutant for the white eyes and gene called miniature, which causes the wings to be very tiny. So a white-eyed miniature wing female crossed to wild-type male. The females that we generate from this, the F1, are going to be all wild-type, and the males are going to be mutant. And so they're going to have an X chromosome with the 2 mutations on it, or a Y chromosome. And so we can just look at crossing over or independent assortment in this female, and the male is only contributing either a Y or mutant alleles. And so when we look at the F2, we can look at all the offspring, and it will give us a good mapping for this pair of genes. So here's a parental type and a parental type among the progeny. So the 2 wild types with the 2 mutants. Notice there's approximately equal numbers of males, similar numbers of males and females. And then there are the recombinant types, and there's quite a few of them, but they are smaller in frequency than the parental types. And we can calculate a map distance, and it's okay to just include all the data here, add up all the recombinants and divide by the total. So 900 divided by 2441, which gives us 36.9 map units. Okay. And there's, the key here is there's no frequency, there's no difference between the males and the females in this case. And you could do this same thing with 3 genes, with the non-crossover, the single crossover, and the double crossover, etc. categories. So you could do all the same analysis. Now, but if we'd done the cross the other way, we can still analyze the data, but we just have to be careful. So suppose we took females that were wild type, homozygous, and males that were mutant. We still have a dihybrid female here, or we could do this with a trihybrid. But the males, in this case, have the wild type chromosome that they inherited from their mother. So if we didn't go and do a test cross, but we just allowed the F1 to breed with each other, this is not going to be a test cross. And so when we look at the progeny, what we're going to see is that all the females get this wild type chromosome from their father, so they're all going to be wild type. And their other chromosome can have crossovers and stuff, but we can't tell. We don't know what's going on with the other chromosome on these females. But the males only get whatever X chromosome their mothers have. So there are parental ones that are passed on, and then there are recombinant ones that are passed on. And because the males are getting the Y chromosome from dad, we can tell from their phenotypes what their genotype was and which chromosome they got from their mother. And so this would be the way we would do the experiment. We would only count the progeny from that were male, and we would discard. We would not look at the data from the females because this isn't going to help us, right? And if we added all the data together and only counted these, we would totally get the wrong recombination distance because we're missing whatever recombinants these females had. So we always look at just the males for that. So the hints are if you have females that are heterozygous for 2 genes or 3, you could do it with 3 genes, mated to males that have only the recessive alleles, you can use all the progeny, add the males and females together, there won't be any difference. But if the females are crossed to wild-type males for some reason, then only the male progeny are going to show you the correct results of the crossing over in that female parent. And so you're going to ignore the data from the female offspring because they will skew the numbers very badly. Now, you can use these R values to draw maps, but if you're given a genetic map or an R value, you can actually make a prediction, right? So suppose someone said, okay, vestigial and purple are linked, and they're 13 map units apart. So if we were going to do a test cross, what would we expect to see, right? And if you were given the genotypes, okay, suppose you had a dihybrid that had this genotype, whoops, not R plus, and then like this, and we did we crossed it to a tester male, say. Then you could figure out, okay, what proportion do I expect to be wild type for both or mutant for both? And what proportion do I expect to be mutant for one or mutant for the other? These are the crossover ones, right? So if I know that there should be 13% crossing over between vestigial and purple, then I know that half of them are going to be this type and the other half are going to be this type. So I'm going to expect 6.5% each of these. And then the rest of the chromosomes are going to be non-crossover chromosomes. And so that will be 87%, 100 minus 13%, and half each of these. So that I'm expecting to see 43.5% for each of these. And a good way to check and make sure that your numbers have been correctly calculated, they should add up to 100%. So that's always the best check. Now notice that, of course, you're not going to get exactly 43.5%. And when you look at the actual results of the crosses that we talked about in the previous, we've talked about vestigial and purple a few times, they're never going to be exactly the same. But this is what we would predict in theory. And then, if we got 22%, 21%, 38%, and whatever the rest is, what is that? 81, so 19%. Or if we get numbers that are weird like this, then we know something is wrong, right? So this is our theoretical prediction, but we can go in either direction. Look at the numbers and figure out what R is, or knowing what R is, we're doing an experiment. What progeny do we expect to see of each type? And then we can compare the actual results that we get and see how close they come to that reality. So this is, basically what we would expect. Okay, now, so that concludes sort of the mapping part. We'll have lots of practice in the class. And what we'll move on next is what is sometimes called non-Mendelian genes, which is just not right, because genes are Mendelian. Genes always segregate the way Mendel predicted. but sometimes there are complications. And so we're going to look at some cases where, for example, you don't have simple dominance recessiveness, or you don't have just one dominant and one recessive allele, a whole bunch of different alleles that have a complicated relationship in how they work and so on. Ones that are only expressed in males, even though males and females have the alleles and so on. So there's those kinds of different situations, and we're going to investigate a whole bunch of those kinds of complications next time.